Okay, welcome. Good morning, good afternoon, good evening, wherever you're joining us from around the world. It is 12 o'clock on the hour, so we're going to get started here in the UK. So welcome to you all. My name is Professor Benedict Michael. I'm a consultant neurologist and director of the Infection Neuroscience Lab and co-lead on the COVID Clinical Neuroscience Study, or COVID-CNS. It's an absolute pleasure to have all of you with us today for what is due to be a really exciting update over the next hour or so of the findings, of the nature of what we've been doing, and also the findings of the COVID Clinical Neuroscience Study. It's really, really great to have so many of you registered and so many of you attend. I know that many of you are actually participants in the COVID-CNS study or have been otherwise involved. So it's really, really great to have your involvement and to be able to feedback a little bit of what we've been doing. So the format is effectively, I will give a very short 5 minutes or so introduction into the background and why we did the study. And then we'll hear updates from each of the working groups in the COVID-CNS study about the work that they've been doing. Okay, so I'm sure none of us want to remember too much of 2020, but if I can take us back briefly to 2020 and just remember how we felt at that time as the newspapers started to report handfuls of cases of COVID-19 occurring in the UK. Some newspapers, of course, handling our concerns with a bit better tact than others. But of course, for those of us in the neuroscience community, the introduction of COVID-19 to our shores and around the world was not anything new. It was in fact back in 1918 when this gentleman you can see here, whose name is Dr. Konstantin von Economo, who was an Austrian neuropathologist, did absolutely groundbreaking work about how the Spanish flu pandemic could impact the central nervous system. And ever since that time in 1918, we as an academic and clinical neuroscience community have been befuddled by what is causing this. To what extent is it these pandemic viruses actually getting into the central nervous system? Is it perhaps not the virus, but immune cells getting into the nervous system? Or actually, is there something about the brain itself and the way it reacts to infection outside of itself that causes brain problems? Now, very early on, our biggest concerns were, could the virus cross what we call the blood-brain barrier and enter the brain? Did the virus not get into the brain, but somehow impact the blood vessels, the arteries and veins of the brain, and therefore cause brain dysfunction? Or was it something about the immune system? Was it these infectious proteins we call cytokines that were causing a storm inflaming the brain? Or was it actually that the virus was gone, but the patient's antibodies to the virus were actually affecting the nervous system? And that's why really about February in the 2020 in the pandemic, we set up a surveillance study called the CoroNerve Study Group, which was basically a collection of the UK's major clinical neuroscience bodies for neurologists, stroke doctors, psychiatrists, so that they could tell us about the sorts of cases they were seeing. And what happened when we had this notification system was that we started to see cases not just of COVID through the UK, but also people being hospitalized with brain complications from their COVID, just as much as they were being hospitalized with COVID itself. We published these data pretty early on in 2020, and we showed that it was indeed blood vessel complications that were occurring in lots of patients, but also this thing we called altered mental status, which is like a delirium where the brain is not functioning properly in response to infection. And what we were able to do with this sort of surveillance technique was track how these cases were occurring. So the light gray shows you hospitalized patients with COVID in that first wave. The dark black line shows you patients with blood vessel problems like strokes. And the dashed line shows you patients with alterations in their brain function, but not due to stroke. And these early data that actually brain complications, whether we call them neurological or psychiatric or a combination of both, were actually occurring in lots of people who were hospitalized with COVID. And in fact, they were actually occurring in some people who had no chest symptoms, so no respiratory symptoms like shortness of breath with their COVID, but they just had neurological manifestations. And it was these findings that, working closely with the World Health Organization, helped to support screening criteria for COVID in these sorts of patients. Of course, this was a global pandemic, so we took a really international view to what we were finding in the UK. And when we compared our study findings in the UK to just over 80 studies around the world with about 2,000 or so patients, we did find these altered mental status or delirium patients, or what we call in the sort of neurobiology of this, we call encephalopathy, which is a fancy way of saying brain dysfunction, was occurring in a lot of these cases. And what we also found was it wasn't just adults, it was predominantly adults, but it wasn't just adults, it was also children that were getting some of these complications. We tried our hardest at the time to try and develop guidelines to help doctors distinguish this brain dysfunction from common things like lack of oxygen or problems with your salt levels or liver or kidney function, so problems outside of the brain having an impact on the brain, from those problems where there was actually a problem in the brain itself, like inflammation, like encephalitis. And it was really clear by this point that these acute, severe neurological complications were occurring. They were often occurring in people who were healthy before COVID, but we really didn't understand the biology, the mechanisms underlying how this was happening. And we didn't really know how to detect it by looking at what we call biomarkers, so proteins in the blood that tell us a bit about how the patient's doing and what might be happening in the brain. But what we did know was that this was really, really important to get to the bottom of, because if people have a brain disorder like encephalitis or demyelination or stroke because of COVID, this potentially could result in lifelong disability. And that's why we were delighted when the Medical Research Council funded us to run the COVID-CNS study to look at these clinical features, symptoms and signs, but also the underlying biological mechanisms, trying to find out who was at risk and what the consequences, the sequelae were. And our aims were really to look at these biomarkers in blood, try and get at the underlying biology. And our hypothesis was really that there was some combination of brain inflammation, brain injury, and maybe genetic risk that would tell us something about the biology that was driving these complications and their neurological and psychological consequences. So we had just over 180 people in the consortium in the end. We hoped to cover all of the UK. Unfortunately, the ethical requirements for Scotland and Ireland made that too slow, but we did manage to cover all the major neuroscience hospitals in England and Wales. And we did manage to recruit to target just over 800 patients, about half who had COVID with lung problems and about half who had COVID but brain complications. And we compared our data constantly to that we were seeing through our global COVID Neuro Research Coalition. I must draw things to a close in the introduction by thanking all the great teams around the UK, just a few examples here, who were involved in recruiting patients and particularly to the patients themselves. We identified patients, whether they were discharged from hospital or whether they were in hospital. They were consented. We took a series of blood samples. We took some information about how they were doing in hospital. We saw them after discharge from hospital for an examination and to assess their thinking, their cognition and their mental health, and then followed them up online for later studies. And about half of the patients actually got a brain scan as well. And now what we're going to do over the course of the next 55 minutes or so is hear from each of the working groups that have been working on different aspects of the neurological and psychiatric complications of COVID-19. And that includes the neurology, the neuropsychiatry and genetics, the neuroimaging, the brain scans, these biomarkers and immune responses in the blood, and teams looking at the cognition or the thinking of patients who've had these complications of COVID. And lastly, and most importantly, we're going to hear from Dr. Ava Easton, who's Chief Executive of the Encephalitis Society, about the really transformative work that we've been doing in collaboration really throughout the COVID pandemic and particularly throughout the COVID-CNS study to make sure we're, engaging with patients, recruiting patients, but crucially doing research that matters to patients and taking opportunities like this to feed back the findings of our research. Thank you to all of you for attending. Fantastic attendance, over 100 people, which is wonderful. Thank you particularly to the COVID-CNS participants who are joining us on this call for all of your support and for all of your involvement. And now to hand you over to a combination of video interviews and live interviews from each of the working groups. The first of which will be from the neurology working group. So it became apparent really quite early on in the pandemic, early back in 2020, that although many people were being infected with SARS-CoV-2 and developing COVID-19 and having respiratory symptoms, there was this subgroup who were developing neurological symptoms of COVID. So what we set out to do in the neurology working group of COVID-CNS was really to identify the clinical characteristics, so the symptoms and the signs and the demographics of these people, and the results that we were getting in their blood results and their brain imaging, really to try and understand who was at risk of developing neurological complications of COVID-19 and what those neurological complications were.
My name is Daniel van Wamelen, always a tricky surname. I'm a clinical lecturer at King's College London. So my main interests revolve around movement disorders such as Parkinson's disease, Huntington's disease. I'm currently working mainly on sort of biomarker studies, neuroimaging, but also clinical sort of phenotyping, so really kind of digging into the clinical details of people with these conditions.
Yeah, so one of the first things we had to do was make sure that our research was relevant to the international community. Of course, this was a global pandemic. So the first thing we did was develop what we call clinical case definitions. So we published those early in 2020, but to validate them internationally, we worked closely with the World Federation of Neurology to evaluate these case definitions using 12 case scenarios, anonymized, and we got involvement from about 150 different doctors of all sorts of different specialties for around about 45 different countries around the globe, including the global south and low and middle income settings. And it helped us validate that what we thought we were seeing really was applicable in the global context.
The main thing we're currently working on is, given my background, is movement disorders. There have been reports whereby COVID-19 infection seems to be a trigger for this type of disorders, where especially there have been reports of people developing Parkinson's disease following a COVID-19 infection, or at least symptoms that look like Parkinson's disease. And what we don't quite understand is how that works, so what are the mechanisms behind that, what is causing people to develop this, and also, to be honest, if this is actually something that's truly there.
The main thing that we found was that although strokes were common, which was well recognized, the next most common complication of COVID was an alteration in mental status. But what we were also seeing was this younger group of people who were getting really quite confused with COVID. And the next question, of course, was why, and what does this mean for their outcome?
The first step we've done is really to look in the big cohort of people who've participated in COVID-CNS, really to see who has developed a movement disorder. So we've had over 800 people participating in the study, which I think is really just underlines a really great number. And out of those sort of 800 people, we have identified 61 who fulfilled those criteria for movement disorders. And I think one of the interesting things we found, and we definitely need to explore further why that is we observed that it seems to be mainly people coming from a Black background, so people from Black ethnicity, really from that BAME group of participants.
So there's two things which are there's lots to do, but there are two immediate next steps. Number one is we want to look at these people, young people with altered mental status. We want to look at their clinical features. We want to look at their brain imaging. And crucially, we want to look at their immune system to try and better understand if there are antibodies that are causing this problem. And the second thing we want to do is understand really what does this mean for the longer term. But we really want to know, what does this mean now for people in terms of their independence, whether that comes to activities of daily living like self-care, or whether it comes to things like employment and education. And ultimately, crucially, what does this mean for the aging brain?
So one of the things we're trying to really help us, make that translation into clinical practice is we also want to see, we're following up people now as part of the study, really to see are any of these changes we've seen, let's say, have some Parkinson's symptoms or have, those kind of those jerky movements. Is that something that's, let's say there forever, they've developed it now and it continues to stay that way? Or is it something that's transient, that goes away over time?
For all of the devastating tragedy that COVID brought, it has represented a unique natural experiment, if you will, where a whole population has been exposed to the same virus at the same time, which allows us an opportunity to understand this. And this is not our first pandemic, and it's certainly not going to be our last. So understanding this for COVID has huge implications for preparedness for the next pandemic, and perhaps even for seasonal infections like flu. Marvellous. Thank you very much. Seems strange to thank myself, but thanks to Dr. Dan Vallerman, who was calling videoing in from KCL. So we will now be handing over for an interview with the Cognitive Working Group with Dr. Ava Easton and Dr. Greta Wood. Before I do, I should have said at the beginning, I was remiss to, you'll see a Q&A button, looks like two speech bubbles at the bottom of your screen. Do please feel free to pop any questions you've got in there, questions or comments, throughout the course of all of these interviews and videos. And as the panel, we'll try and get to as many of them as we can at the end of the presentations. Thank you. Over to you. Thank you, Cognitive.
Many thanks, Ben. So this is the neurocognition element with Dr. Greta Wood. But first of all, I understand that we have an audience poll to launch. So if we could launch that poll and then see the results of that, please, first. So you should be able to see the poll on your screens now. If you could all choose one of the answers. And then hopefully we should be able to see the results of that poll in a second or two, please. Do we have the results, please, of this poll? Let's have a look and see. So people, we have a majority, 91% think that COVID-19 has a lasting impact on cognition, and 9% disagree with that. So that's really interesting. Thank you, everybody, for participating. If we could close the poll down now, I'm going to turn to Dr. Greta Wood. And Greta, I've got three primary questions for you. But I think, first of all, let's start at the beginning. Can you tell us about what the Neurocognition Working Group have been researching about within this study?
Yes, absolutely. So the question we've put to the audience is the same question that we really started to try and answer. So does COVID impact cognition, and does it have a lasting impact? And I think now that there is a general opinion that COVID has an impact on memory and thinking, but there are big gaps in the research. So most studies exclude patients with brain complications. They don't look at blood markers, they don't look at brain imaging, and they don't follow people up over time. And these gaps in the research really make it very difficult for us to support and to treat patients who have problems with their cognition after COVID. And so in COVID-CNS, what we've set out to do is to look at cognition, but also blood markers and brain imaging, bring together expertise in those different areas and fill those gaps.
Thank you, Dr. Wood. Those are really important gaps. Thank you for highlighting them. So let's cut to the chase, I guess, really. What are the findings so far?
Yeah, so thank you for people who participated in COVID-CNS, and because of that we've learned so much, but I will focus on the things that I think are the most important. So firstly, 1 year after COVID-19, we've seen that there are significant deficits in people's cognition, so their thinking and their memory. So in people who've had COVID without brain complications, we've seen that those deficits are equivalent to about 20 years of aging. And the people that seem to have the biggest problems with their memory are people who were unwell early in the pandemic, who had more severe COVID-19, and who have symptoms of depression. And then in patients who've had COVID and a brain complication, we find the most common complication of COVID on the brain is encephalopathy. So as mentioned earlier, this altered mental status or confusion. And it's generally thought that that's short term, but what we're seeing is that 1 year after COVID-19, patients who had encephalopathy when they had COVID or when they were unwell with COVID, their deficits in cognition were equivalent to about 40 years of aging. So really significant problems for those patients. And then when we look at their brain injury biomarkers and their neuroimaging, what we're seeing is that, again, 1 year after COVID, there are raised brain injury biomarkers and reduced grey matter volume in patients who've had COVID, especially in those who've had brain complications, and that reduced grey matter volume is worse in those who have greater problems with their memory.
Well, thank you for that fascinating update, Dr. Wood. Looking to the future, what are the group's plans? And I guess, very importantly, what does that mean for patients and the public, all of whom really have been affected by this pandemic?
Yeah, absolutely. So firstly, in terms of future plans, so far what we've done is kind of really in-depth look at cognition and then a focused look at the brain imaging. And so what I'd like to do now is do that in reverse. So have a really in-depth look at those brain images, not just the structure, but also the function and the activity of the brain and what that means for people's thinking and their cognition. And also slightly change our approach. So far we've been taking a kind of traditional, what we call a hypothesis-driven approach. So we have an idea and then we ask, is it right? And I think I'd like to take a sort of newer approaches, what we call data-driven approaches. So instead of saying, this is our idea, is it right? We say, without any assumptions at all, what's the data telling us? And that can sometimes give us new ideas or new insights, which is really exciting. And from a patient's perspective, what I think is really interesting is that these patients knew they had problems with their memory and their thinking. In fact, when we asked the participants in COVID-CNS, are you concerned about your memory? If they said yes, 9 out of 10, their cognitive scores would be less good than we would expect, and 3/4 would have a significant deficit. And so as clinicians, it's very important that we listen and that we believe. And I think for the patients, if they do have concerns about their memory, they can go to their GP, they can get an assessment. And I think finally, the other thing is, I think we need to think about our brain health a bit like we think about our heart health or other things. There are lots of things we can do to keep our brains healthy, which actually are a lot of the same things that keep our heart healthy. So we can stay active, good diet, reduce alcohol, and particularly for our brains to stay socially active.
Dr. Wood, that was a fascinating insight in the very short space of time that we have. Thank you so much for joining the webinar today. Thank you to you and all of your team at COVID-CNS for the incredible work that they've done. And I'll hand back to you, Professor Michael. Thank you.
Marvellous. Thanks very much, Dr. Rose, and thanks, Dr. Wood. That was great. So we'll now be teeing up the video for the neuroimaging working group.
Hello everyone, my name is Bethany Faso and I'm a PhD student at the University of Liverpool, and this is Brendan Sargent, who's a clinical research fellow, and we're part of the group for the neuroimaging working group. And to start us off, Brendan, who actually is the neuroimaging working group?
The neuroimaging working group is a group of individuals as a subgroup of the COVID-CNS consortium, and they use brain scans in the context of COVID illness as part of research. So it's a multidisciplinary team made up of experts and trainees from a variety of backgrounds and from sites across the UK. So for example, you can see we've got a number of clinicians such as psychiatrists on the screen, neuroanaesthetists on the screen, neurologists, and trainee doctors as well. We also have imaging neuroscientists who are experts in research in the context of brain scans, and we also have clinical neuroscientists as well. So we have a full breadth of the spectrum from brain scan all the way to clinical application. And these individuals are from across the UK, places like Liverpool, Cambridge and London. So Beth, what are the aims and the contributions of this imaging group?
So the sweeping kind of overall aims, I would say, is to investigate the structural and functional brain changes associated with the neurological and psychiatric symptoms of COVID-19. Now, I'd say this we can put this down to three kind of main areas. The first is harmonisation, and this is where we want to get the same imaging parameters for all these different sites. A paper released last year shows here the 4 different of our main sites, Oxford, Cambridge, KCL and Liverpool, called travelling heads, where we want to optimise the imaging outputs so we can have consistency across sites. The second is we want the creation and integration of imaging variables. So when we're processing the images to create the outputs, which can be integrated into the larger database, we want these to be readily accessible for other working groups to be able to extract and use. And lastly, it's the diagnosis, mechanisms of change and therapies, and really want to use these metrics to further understand these three areas.
The group has utilised lots of different imaging modalities, and as Beth said, the scans are harmonised across sites so that the scans are comparable between individuals. And an example of a scan, one of these modalities, are the structural scans seen here, and that allows us to look at areas of the brain and the changes in those areas in terms of their structure and shape. For example, the cortex, the layer on the outside of the brain. Another example is functional magnetic resonance imaging, or functional MRI, which allows us to look at the activity in specific brain regions. And then there's other forms of MRI as well, such as specialist scans that let us explore things like blood flow in the brain or the white matter connections between different regions of the brain. So all of these different types of scans are processed in order to produce image-derived phenotypes, which are essentially numbers or metrics that represent a biologically meaningful measure of structure or function, so that you can take those scans and turn it into something meaningful and clinically relevant for research.
So there's 4 sections to this with our future plans. The first is what ongoing projects do we currently have? So we've got a few projects which are researching cognition, stroke, encephalitis and movement disorders, and what the imaging can contribute to that. And we're underway with many more projects on top of that, which are currently in the pipeline. The future, we also want to have more collaborations. We want to work closely with the other groups where we can pool lots of data and other experts. Third is pre-processing the imaging further. Some of the more advanced sequencing, such as arterial spin labeling and SWI, take a bit more processing, but will be really vital for future work. And lastly is the scientific brainstorming that we're really moving towards now as well, where we want to work together to come up with the most pertinent research questions. So we regularly discuss these possible ideas in our groups.
Brilliant. Thanks to Brendan and Beth there for giving us a brief update on the neuroimaging working group. We'll be turning over back now to Dr. Easton for another interview, this time with Dr. Cordelia Dunai, who's a postdoctoral research fellow. Again, just a quick reminder about questions coming in. We've got lots of great questions coming in the Q&A, but please do feel free to pop your questions in now. Don't hide your light under a bushel or sit on your hands. Do let us know what your questions are, and we'll do our very best to answer them after the presentations or after the whole webinar. Thank you, Dr. Easton, over to you.
Thank you, Professor Michael. Dr. Dunai, thank you so much for joining us. As Professor Michael said, you're in the biomarkers and immunology working group. So I think, first of all, let's start with the basics. Can you explain what biomarkers are and which ones you studied?
Yes, for sure. Thanks for having me. So biomarkers are molecules that can be measured reliably and are associated with particular diseases, and they can change in response to therapy. So they're very useful for tracking disease progression. A lot of times biomarkers are in the blood because that's an easy thing to sample, and that's what we studied. We studied biomarkers that are present in the serum, which is a component of the blood. And they fall into 2 major categories that I'll go over. So first of all, cytokines, which are proteins that the immune system uses to communicate. So they can be pro-inflammatory, for example, or anti-inflammatory, excuse me. And the second category is brain injury markers. So as the name suggests, these are proteins that are highly expressed in the brain and nervous tissue. And so under healthy conditions, they'd be at very low levels in the blood. And if you can detect them at high levels, that's a sign of brain injury.
Thank you. Can you tell us a little bit more about what the role of the immune system is in COVID-19 infection?
Yes, so I'm a immunologist, so this is my favorite subject. And obviously, the immune system has a very important role in protecting against viruses. It has a lot of different specialized cell types and proteins that are involved in clearing the virus and also building a memory response. So if you're exposed to the same virus again, you can clear the virus. However, sometimes there's an overreaction of the immune system, and this could cause tissue damage itself when it's not regulated properly. So that's immune-mediated pathology that we're very interested in studying. And actually, there's several pro-inflammatory cytokines and clotting factors that have been associated with COVID severity, and in particular, neurological complications. So that's why we wanted to study those.
Thank you so much. Well, let's turn to the crux of this, and I think you've got a slide that you'd like to appear for this question. But what are the findings so far from the biomarkers and immunology working group?
Yeah, so just to reiterate a little bit of background that Ben introduced, we know from autopsy studies that it's the minority of cases of COVID-19 where there's viral presence in the brain. So that leads us to hypothesize that it must be indirect effects of the virus most of the time causing neurological complications. So, for example, immune activation could be affecting the brain. And so, as introduced in the COVID-CNS study, the marvelous thing about this is that we have the groups to compare these biomarkers. So we looked at COVID negative controls versus COVID positive controls. So that's patients that had COVID but didn't experience any neurological complications. And then the third group of COVID-19 patients with neurological complications, including strokes, encephalitis, seizures, movement disorders, a whole suite of conditions that we can stratify by. And this slide is an overview of the key findings. So again, we measured these different biomarkers in the blood, brain injury markers, autoantibodies, pro-inflammatory cytokines. And we could look at acute cohort versus the COVID-CNS cohort, which is a lot of times we have samples from months to even beyond a year after initial COVID infection. And we see a lot of, so focusing on that group of COVID neurological cases, we see elevated biomarkers early on. And then really importantly, even months to more than a year after COVID infection, we're still detecting high levels of brain injury markers. So this could be because of ongoing brain injury, and we really want to understand that more. And it was a key signal that we found. We kind of predicted if there's ongoing inflammation, we might detect high levels of pro-inflammatory cytokines, but we don't see that in the blood. But that doesn't rule out that there's still local inflammation going on.
Thank you. Well, that probably leads to my final question for you, which, as I asked Dr. Wood, what are the future directions of the study in terms of biomarkers and immunology?
Yeah, so far we've looked at 98 COVID-CNS participants, and we've written up a research paper on this with the whole group led by Professor David Menon, who's here today, and Leonie Tams at KCL. And so we've got that paper coming out soon in the journal Nature Communications, but we also have almost totally completed analysis of all over 800 COVID-CNS participants. So we're able to do more in-depth analyses of the biomarkers there. And what I'm really excited to see is with these larger numbers, being able to stratify by the different conditions, for example, encephalitis versus a movement disorder, seeing if they have a unique immune signature that we can pull out. And then, of course, we're a molecular biology lab too. So we're working on in vitro blood-brain barrier models to understand how SARS-CoV-2 affects brain cells. And basically, our aim is to understand what pathways are involved and then use therapies to block that and study how we can protect the brain cells.
Well, again, thank you so much for joining the webinar today, and to you and the working group for all of your fantastic work. And I'll hand back to Professor Michael now. Thank you.
Marvelous. Thank you. That was great. A very complex area covered very clearly, I think, but happy to take more questions. So do pop in the chat. Loads of great questions coming in. Please feel free to add extra questions in the Q&A box at the bottom. We'll be moving on now to a video interview for the Psychiatry and Genomics Working Group. Yeah, as I say, please do pop questions in as you like. Thank you.
Hi, my name's Tim Nicholson. I'm a reader in neuropsychiatry at the Institute of Psychiatry, Psychology and Neuroscience at King's College London.
Hi, my name is Tom Pollack. I'm a senior clinical lecturer in neuropsychiatry at the Institute of Psychiatry, Psychology and Neuroscience in King's College London. One of the reasons this came about and one of the areas of real interest for us was there's been this long established relationship between infections and neuropsychiatric problems, mental health problems. And some of this evidence comes from big population studies showing that people who have an infection are more likely to develop mental health problems of really any different kind. And so in the early days of the COVID pandemic, when we realized that this was going to become a worldwide problem, there was some real concern that there may be some very serious and very widespread mental health problems occurring as a result of the pandemic. We teamed up with Professor Ben Michael and colleagues working on the coronerve study, and we were able to get an early signal that people who have been infected with COVID did, in some occasions, develop mental health problems. But it wasn't entirely clear from these early studies whether we could be confident that it was the COVID infection that was causing these mental health problems, or whether there may be some other explanation, or whether these problems might just be emerging as part of the general impact and general sort of confusion that occurs when someone has an infection such as COVID.
So the research so far has really looked at the clinical characterization of the types of presentations, the types of symptoms that people get following COVID. The first step has really been to look at the types of symptoms that people present with or clinicians think might be related to their COVID infection, and whether that's occurring in the first few days of the infection or whether it can follow later on. Within psychiatry, we have a much more challenging situation compared to neurology, where we need to define things just on symptoms. We don't have tests or ways of securing a diagnosis based on the brain scan or a blood test. It's really just the cluster of symptoms. So it's a complicated thing for us to try and get to the bottom of. So the main results have been that there's no clear signal of any particular disorders apart from cognitive impairment, so memory problems, and some signal for psychosis, so that's sort of either delusions or hallucinations, which can overlap a bit with two common conditions, sort of delirium and sort of a psychosis, like a schizophrenia-like illness. Those sorts of complications are things that can overlap a bit, and we've really been trying to tease out what the separation of those two common problems, which can occur in the first few days, but also perhaps sometimes a week or two, even a month or two after the initial infection. So the other thing that's been interesting is that the data we've got from this study has been very much in line with the very early data we got from the surveillance studies.
So I think one of the really exciting things, we're at the sort of exciting stage of the data analysis now, is thinking a little bit more mechanistically. And what I mean by that is, what are the causes of the particular neuropsychiatric presentations in these people who have had COVID infection? I think we've touched on the fact that when it comes to mental health symptoms, they're quite complicated, because often there's a variety. There's sort of almost too many causes that could potentially cause a particular symptom. It's difficult to know which one is relevant. So, for example, in the case of depression, low mood, or even paranoia or something, well, of course, it could be the direct effect of the virus on the brain. It could be a more general inflammatory effect of being unwell, which over the last 10 years or so, about brain mechanisms, particularly sort of immunologically mediated brain mechanisms that can give rise to neuropsychiatric symptoms, thinking about things like sort of autoimmunity or inflammation, or even more generally genetic causes, which might explain why some people are more vulnerable to developing neuropsychiatric problems following an infection than others. And we're hopefully going to be able to tease apart these different potential mechanisms and answer the question of which ones are the most relevant in leading to the neuropsychiatric problems after COVID, and in turn, how then we might be able to prevent or cure these kinds of problems in future.
But the great thing about this study is it's brought together psychiatry as part of a whole brain project, so looking at and really uniting the whole of clinical neurosciences in the UK, which is really the first time it's ever really happened, and allowing us to therefore get a bigger picture, particularly at the interface of psychiatry and neurology. A bonus to be working together with all our colleagues across neuroscience, which I think has really set the tone for future work, not just within COVID, but also within future pandemics and important infections. So it's a brilliant project to be part of as far as that is concerned.
Great. Thanks to Tim and Tom from the Neuropsychiatry and Genomics Working Group. We are tight on time, and I'll hand straight over to Philippa Chapman, who's Deputy CEO of the Encephalitis Society, who'll be doing an interview with Dr. Easton about patient and public engagement in the study. Thank you both.
Thank you, Professor Michael. Right, first question then, talking about patient and public involvement, or PPI as it's better known in research. Can you tell us what that actually is, please?
Absolutely. Well, patient and public involvement is about research that's being carried out with or by patients and members of the public, rather than to, about, or for them. So it's about authentically and deeply involving people who might be affected by or concerned about the research in question. So this could be patients, potential patients, carers, and of course people who use health and social care services, which of course is most of us, and people from organisations that represent people who use services. So for, you know, the Encephalitis Society is a great example, and of course patient and public involvement is also about members of the public, and when it's done well, it's a two-way process that really encourages researchers to really listen, to really interact with the people who their research most affects.
Fabulous, thank you. And can you tell us what has been the role of the PPI panel in this actual study? Tell us a little bit more.
Thank you. Yeah, it was extensive, and as Professor Michael said, we're running a little bit out of time, but the role of the PPI panel in this study was really to ensure that the study outcomes were related to patient benefit. In this case, in whom does COVID-19 cause brain problems? Identifying the nature of those brain problems, and as we've heard from Dr. Wood and Dr. Dunai and other colleagues, hypothesizing solutions, such as this might have been clinical care guidelines, identifying patients for targeted clinical trials. So a whole range of things really, but perhaps most importantly, having the aim of ultimately improving patient outcomes at its heart. And as a panel, we helped the study in a whole range of ways, but a couple of examples for, you know, exploring and gathering feedback from people that had been affected by the condition, especially as the study was being set up, what was working, what wasn't working well, providing a patient and public perspective on study discussions, materials and information that was created as part of the study. And of course, now we're in that process about disseminating study findings and creating opportunities like this webinar today, which was very much driven from our patient panel feedback. And I'm thrilled. I can see lots of questions coming in, and I can see that we've been joined today by a lot of people who COVID-19 has affected. So hopefully we're doing the right thing.
I'm conscious of time. Should we finish up by, can you tell us some of the highlights of this study, please?
Oh, well, gosh, there was so many, actually. It's been a genuine pleasure actually working on this study. Recruitment to studies such as the recruitment of patients into a study can often be a real challenge, but this study actually flew through that. So it actually freed up more time to concentrate on other things, such as disseminating the study's findings, as we've just alluded to. So many papers have come out of this study already, and we now have the pleasure of being able to get some real public engagement going, such as this webinar. I have to say, you know, thanks as well to the other patient organisations that, you know, we held hands through this, MQ, the Stroke Association and the Neurological Alliance, who were all aligned with me on the patient and public involvement panel, but also in particular acknowledging the steering committee and the heads of the study, who were genuinely supportive of the patient and public involvement element. So that made our job a lot easier. And that isn't always the case. Sometimes in patient and public involvement, you have to shout to be heard, but that wasn't the case on the COVID-CNS study. We were always a priority. And so that for me was, I think, one of the highlights of the study.
Fabulous, thank you.
Brilliant, thanks both. That was great. And just to echo those words, David, it's been wonderful to have such an engaged patient and public panel. You know, it's not just the scientists that have to be engaged, we all have to be engaged. It's been wonderful. I'd like to invite the remaining panelists to turn their cameras on and unmute if they're able to, and to introduce them. So we have Professor Ed Bullmore and Professor David Menon, both from Cambridge, who lead the neuroimaging and biomarkers groups respectively, Professor Charles Leake, who leads the cognitive working group, and Professor Jerome from Liverpool, and Professor Jerome Breen, who's co-PI with me on the COVID-CNS study, who's in psychiatry and genetics at King's College London. So we've had loads of great questions come in. We'll get to as many of them as we can. I'll try my best to direct them, but others on the panel, do please jump in if you'd like to add. Firstly, just to reassure those who joined a little bit later, this is being recorded and will all be available. So please don't worry if you missed the first 5-10 minutes or so. It will all be recorded and available. So we've had loads of great questions. Of course, I hope those in the audience can appreciate that individual questions about individual patient specific problem is very hard for us to answer when we don't have the full details, but we will do our best to try and answer the questions as broadly as possible. So the first question is around about how do our findings relate to community cases? So this is a question we get a lot. What about the people that weren't hospitalized? What of our findings, or how might our findings help inform what's been happening at a community level? And perhaps, Jerome, you might want to come in on this.
Yes, that's a complex question. It's worthwhile reviewing what the COVID-CNS cohort is. It's incredibly severely affected cases for the most part. It's people at the age of 30 who've suffered a stroke. It's people who've got severe encephalitis, who've had other very severe problems. Generalizing that back to the community in terms of the difficulties experienced by the community is hard. However, I think the value of the data set, though, is in pinpointing the risk factors for developing those extraordinarily severe life-changing complications. And I think it could inform community prevention strategies in terms for future pandemics, in knowing who needs to be shielded, who needs to be a priority for vaccination, etc. I think that's probably where the real utility of this data set will be.
Great, thank you. Another question that's come in is patients, perhaps this is one for Professor David Menon and Professor Ed Bullmore, is thinking about patients who present with a neurological complication in the context of COVID, but then go on to develop a neurological diagnosis. I think we've had examples of isolated syndromes that subsequently get diagnosed with MS, for example. Is this a common problem? How does it relate to COVID, or is it completely unrelated?
So I'll go first because Ed's still muted. So there are certainly incidences of patients who have what are thought to be immunity-related diseases like myasthenia, MS, that kind of a clinical picture during the disease narrative of having had COVID. We do know that the body's immune response becomes a bit dysynchronous and may cause problems with the neurological system during COVID. So the work that Cord had discussed earlier shows this pretty conclusively. Whether this ties up with the firm diagnosis of MS is a little more difficult to know, because we're going to have to wait for a little while and see how the disease evolves in these people. But we know that clinical pictures of immune-related neurological injuries certainly are associated with COVID, and the follow-up of these patients is really important, because as someone said during the discussions earlier, what we need to know is whether these things happen and then gradually get better, or whether they keep people at risk of ongoing symptoms for a long time.
Ed, I don't know if you want to add anything additional to that.
I don't think I have much to add to what David said, except that I suppose without scaremongering, you mentioned von Economo and encephalitis lethargica at the start, Ben, and that was certainly a case where, well, we don't know exactly what the pathogen was in that epidemic, but it did seem to be followed by longer term emergence of neurological problems. So if we assume that what happened at 1918-19 was a viral infection, people tend to think that's the most likely cause. It is at least an example that it's not just acute events that can follow from these infections, but maybe longer term too. I think COVID-CNS by design is really focused, as Jerome has already said, it was really focused on the most acutely unwell people. And I think it's going to give us invaluable insights into what happens in some individuals in the immediate aftermath of a severe coronavirus infection. The longer term shift in risk for autoimmune brain disease or conceivably neurodegenerative disorders, I don't think our design was never set up to do that. We do have NHS linkage for some of the participants, and there is the community cohort that Jerome has reflected on. But I think really to be sure, or to know more precisely whether COVID has actually sort of shifted the needle on risk for long-term neurological disorders, I think that is going to require bigger epidemiological scaled data sets and longer term follow-up. I think it's probably too early to be sure how real that risk is.
Yeah, thank you. And on that longer term point, I'll come now to the cognitive group, so to Dr. Wood and Professor Leek. Just before I do, we've had a question in the chat about vaccine complications. So in the COVID-CNS study, we did recruit patients who had vaccine complications. The numbers were very, very low. And we know now from the epidemiological data that the risk of infection causing a neurological complication is vastly higher than the vaccine causing a complication. But we are studying those small numbers of vaccine complications too. We just didn't have time to talk about that today. Moving on to cognition now, and segueing on from what Ed has said about longer term, we've had lots of questions about longer term cognitive function and what people can do. It's all well and good saying they've got cognitive problems after COVID. What should they do now? Greta, you touched on this a little bit. Questions about, is this irreversible? What strategies can be used for cognitive impairment? And in fact, one person in the chat who's mentioned that using word games and their role as a musician has helped in their cognitive recovery. I wonder, perhaps Dr. Wood and Professor Leek, if you could comment on cognitive recovery strategies, please.
Well, I'll start and then I'll see what Professor Leek, I'm sure he'll have lots to add. But yeah, we have been looking at recovery trajectories. I think the first thing to say is that we were looking at some of the most severely impacted patients. And we were looking at hospitalised patients and patients who'd had brain complications, many of whom became ill early in the pandemic, and many of whom weren't vaccinated before they had COVID. So I think we are really looking at the severe end of the spectrum. That being said, we are seeing evidence of recovery, and we're doing work to try to better understand who will recover and who won't. And we are seeing some signal that raised brain injury biomarkers are associated with a reduced recovery, but the numbers are small, so we really need lots of people to do the follow-up so that we can have more data and have a better analysis of that. In terms of strategies, in terms of treatments, so we're seeing that in the COVID group, so people without brain complications, when you adjust for age, having a vaccination before you became unwell was protective, and being treated with steroids was protective, which indicates this is likely kind of immune mediated, so an indirect impact on the brain. In terms of after COVID, where we're at now, a lot of the things I've suggested aren't specific to COVID, and I suspect that's because those things will be generally true, whether or whatever has caused your cognitive issue. And so things that help the brain are things that help the heart. So being active, physically active, a healthy diet, quit smoking, control blood pressure, reduce alcohol. And particularly we're seeing, actually there's a news article in the BBC today about the role of social interaction in protecting people's brains. So Charles, what would you add?
Can you hear me? Sorry, I was having problems finding my unmute. I would very much agree with everything you've said, Greta. I mean, It's worth bearing in mind, of course, that this research study, we weren't focusing on intervention, we weren't focusing on rehabilitation for cognitive outcomes, and there's a lot yet for us to understand. I mean, there are treatment options available through clinical neurology, neuropsychology services for individuals with cognitive impairment. But I think, you know, we still have quite a bit to learn about the longer term outcomes and the trajectory and predictors of cognitive outcomes. Yeah, look, you know, it's something we still really need to fully grasp and understand with future research.
Thank you both. So I guess the point there is that we have to first define the nature of the problem, which is very much what we're in the process of doing. And then develop targeted strategies for individual patients going forward. I would like to move, as we draw things to a close, back to Dr. Easton. Just before I do, we've had one question about infection in pregnancy and the risk of schizophrenia in later life. We haven't seen any signal yet of later life schizophrenia in children born to mothers who had COVID during pregnancy. Obviously, we'll need big long-term studies to look at that. There have been some suggestions of infection more generally, being associated with schizophrenia later in life. But that's when we look at millions and millions of people and probably isn't pertinent to an individual mother having had COVID with an individual baby and her specific risk. Jerome, quickly, and then we'll come to Ava, if that's all right.
Yeah, just to come in on that. Yes, there was a story in the psychiatric literature of linking influenza infection with schizophrenia risk. However, over time, that signal has diminished, and there's concerns that actually if there is a signal, that it's very, very small. The most robust signal for schizophrenia risk is actually to do with exposure to famine in the first and second trimester and increased risk of schizophrenia. And that is a very strong effect. But actually the infection effect is, if it does exist, is mild. So I wouldn't say that that's a huge concern.
Marvelous. Okay, well, very reassuring for the many pregnant women who would have had COVID during the course of their pregnancy. Dr. Easton, so we've had lots of questions about how research has come together during COVID, how it galvanized people that were otherwise siloed, neurology, psychiatry. People would like to know more about how we go about sharing our sorts of research findings and how they can find out more about COVID-CNS, about our findings going forwards, and about encephalitis and brain infection and brain inflammation more generally.
Oh, huge question. I'll try and be very brief. We do have a podcast, I think, that we recorded with you, Ben, which people can go and watch. That's available from the Encephalitis Society's website, which is encephalitis.info. We will be renewing some of the dissemination materials that will be available on the COVID-CNS website as well. And Ben, can you remind me the COVID-CNS website address?
If you Google COVID-CNS, you'll find us.
COVID-CNS. There's lots of information on there, including a playbook where we have identified in kind of public and lay language some of the key literature that's come out, some of the key findings from this study. We're going to be updating that fairly soon. So there's lots more to come out. The dissemination will continue. And from a patient and public perspective, I sincerely hope that the work of COVID-CNS group is funded and continues, because I think it's absolutely critical and vital, and we've heard today about the fantastic work that's gone on. So thank you to all of the team and all of the working groups for addressing these issues that were, as you started, Ben, vital for people who were often very frightened in the early times of the pandemic, and as we've now seen in the Q&A, are asking lots of questions about their outcomes and the future.
Absolutely. So just to draw things to a close, do please follow us on Twitter or X, as it's now called, if you'd like to hear sort of real-time updates. Wherever possible, we publish our research as preprints, and we always try and make our findings open access. So that means they're not behind a paywall. You don't have to be part of a university to see them. And we always try and publish either webinars or tweets or Spotify interviews or YouTube videos about what we've been finding. to keep you up to date as new findings are coming out, and there's much more to come out just in the next few months and probably for years and years to come. Thank you to all of the panelists, thanks to all of our speakers, thanks to everyone that recruited patients to COVID-CNS, but crucially, thanks to the COVID-CNS participants, without which there wouldn't be a COVID-CNS study, and we wouldn't be making these real milestones in improving our understanding of the brain complications of infection across the brain-mind spectrum. And thank you, all of you who joined us. This will be available as a video online, and we hope to hear from you again in the future. Thanks again.